So yeah, it's happy 11th birthday, everybody. So I hope there's cake coming up, Ruth. But actually, I think about this today, and I thought this meeting is a perfect example to me of sort of my reflection on what SHIVERS is and how sort of if you plant people around it, things will grow. So when we first started with this, it was, I think, Paul and Aubrey said, you know, they're going to do a Well Kiwis infant trip site visit to New Zealand, Wellington. You want to come? I said, sure. And Ruth followed that up quickly with a little warning that there was a red-headed Englishman with a guitar who was going to be in town. So quickly get on board and get your accommodation booked. So we did that. That was followed a little bit later by an email from Sue. I actually thought, why don't we sort of expand that? And a day early, we'll put another day on it. We'll do a sort of celebration of SHIVERS. And that was good. It was followed a little bit further on the next day, or a bit longer, by something. Well, Nikki thought, because we have some people here, let's do a public lecture. And that was good. And then followed a little bit later by Sue said, well, while you're here, let's chat to Joseph and see what else from the animal side doing. So that sort of turned in from a 2-day thing into a week-long. I changed my accommodation 3 times. But again, I think it's a really good idea. If we have sort of the right things grow. And again, I think it's just a great sort of illustration of why I've enjoyed being part of this for the past 11 years. But what I want to give it very briefly here for the next 8 minutes and 30 seconds, Ruth, is to put a little more bones around, meat around the bones, and sort of exactly from my perspective, a little bit sort of following on what Michael said about sort of the CEIRS program and how at least SHIVERS-II and SHIVERS-IV fit into that sort of the overall program from not so much a NIAID perspective, but from one of the centres within the NIAID network. And there are essentially 2. Perfect. So the first is sort of the CEIRS. So this was the program that really put some of the funds in place to do SHIVERS-II. And we'll come to a little bit about where that came from. And then sort of we moved on to St. Jude CEIRR. Actually, sort of a very biased history of this program. The CEIRS program actually started, I think, with Rob Webster, who of course many of you are familiar with, another Kiwi who, back in '97, there was an outbreak of highly pathogenic H5 in Hong Kong. Also 18 people infected, about a third of those succumbed to the infection. So he put a grant into NIH to study that. At the time, there wasn't much going on in the portfolio of NIH in this area. And so he got it, it was successful. They thought about it, but well, we're going to fund this, not perhaps in this traditional grant mechanism, but actually in a contract mechanism. And that allows a little bit more flexibility to do stuff within it. And that it was funded for 7 years, and it was sort of so successful at the end of that, it provided a lot of information that this is where the CEIRS network was born. So it went from sort of one centre that Rob Webster had to, I think it was 5, that went for 7 years. We recompeted for that, and we're successful. For the next iteration of that, St. Jude CEIRS, and again, we're now into sort of the 3rd iteration of CEIRS, which is now CEIRR. And this is sort of what funds the SHIVERS-IV platform. So, where did all of the SHIVERS come into this? SHIVERS-II really came about from, I guess we can thank the Canadians a little bit for this. So they put out a few publications not long after the 2009 pandemic suggesting that the people who responded poorly to vaccination actually were overrepresented by people who had received vaccination the year before. And of course, from a public health messaging perspective, and someone like Nikki, that doesn't go very well. If you actually get vaccination this year, you're not going to do so well to the vaccine the year after. And NIAID became interested in this and put out sort of a call for, what do we got? We've got a little bit of money to study this, what proposals are out there. And it was good timing for SHIVERS because we'd sort of bled the Mark Lane's wallet dry at that stage. So the CDC project was sort of winding up, or a lot of it. And so it's pretty clear that the platform that was developed from that initial CDC funding was pretty great place to start this. And so hence SHIVERS-II was born, and again, we know the reasons why it's been successful. We're not going to go into that. Where are we actually with that? And so I should say that it's been so successful, and so many of you intimately involved in this know that SHIVERS-II, as we've moved into SHIVERS-IV, we've gone from individuals to households, and there's many individuals within SHIVERS-II who couldn't take part in SHIVERS-4 because of lack of households, I guess, is the main reason. And for still hard to find it true, but Sue told me there's many people in there who were quite upset they were going to get dropped from the study. And so, this is a group of people now that are 5 years into this. We've been collecting samples from them. And so we've been lucky actually to get a bit more money now to extend this for at least a couple more years. So that's great news as well. And thanks Michael and his team for that. So where are we now? And look, this is the only data slide I've got. What we've started to do is break that down. And so we're asking this question. Most of the studies that were done before really looked at the previous year. What happened the previous year and how did that impact vaccination? Of course, the power of the longitudinal cohort is that we don't just have 1 year, we have these 5 years. And sort of this is sort of Tim's breakthrough brainwave here. Sort of if we look at the last 5 years and just a 0 for unvaccinated or a 1 for vaccinated. And we've been working with Henry Wan at the University of Missouri looking at this, who's been developing some learning models around this data. So, what can we learn from not just a previous year, but the previous 5 years vaccination and how that influences. And it comes out that it's actually, as you might suspect, a little complex. It's probably different depending on actual antigens in the vaccine as well. So certainly if you look at the H1N1 here, and just low responders here, high responders here, and this is their history, it kind of looks at like it's more just number of vaccinations you've had in the past that impacts sort of the H1N1, whereas the H3, maybe it's a little more focused to actually that previous year. And maybe this makes sense because we know the H1 is a little more stable antigen in the vaccine. It doesn't change as much. And that's, again, something we're trying to build into these models now, is some of the antigenic change and how that impacts. So, we are moving along with a lot of this, and I think it's going to tell us a lot about the vaccine. I don't want you to take this, again, as being lots of vaccine is bad, because this is really just looking at the response to vaccine. And we know that if you receive vaccine enough, you eventually reach this sort of plateau. So this is how you respond to vaccine, not necessarily how protected you are, but that's kind of where we want to move to now. So we're learning a lot about that. SHIVERS-4, so when it came time to recompete our CEIRS into the CEIRR, as Michael just showed, part of that call from NIH said you had to have longitudinal cohorts in that. And so Aubrey, who's over here, you're going to hear from again, another sort of major partner in our centre, and Sue got together and thought, well, how are we going to do this going forward? Now's the time to sort of refocus perhaps some of the activities we had, and then this is where they sort of come up with this household transmission cohort dynamics that we have now. And so that came about from that. This CEIRR program, unless we do something really stupid over the next few years, is funded for, I guess, another 6 years. So again, we have this real powerful sort of longer term funding commitment to these cohorts, which is great. And this is sort of where that fits into our current St. Jude CEIRR program, looking at sort of overall influenza research and response, pandemic preparedness, interface studies, risk assessment. We heard Paul talk yesterday about the immunological aspects that he's studying. So again, he's using a lot of the samples that are coming from Sue and Aubrey's cohorts to understand from an immunological perspective how that's happening. We're certainly looking for the virologic component as well. So it fits into sort of a much wider program that we're involved in, addressing sort of this overall preparedness and response to flu. I don't think we're going to go through this too much, but this is kind of the objectives that we have for the two programs, but I think we can talk about that later if we need to. But that's kind of where it fits into. Again, I guess apart from Paul Thomas, this is the most famous Memphian. So we want to thank everybody for doing this. I always sort of sit there and I get a lot of the glory from how successful some of these cohorts are, but I've never run a cohort in my life. So, thank you everybody, Sue and sort of the whole team, and of course the people behind me who actually take part in this. I still find it incredible that after 5 years of getting blood drawn, people still want to be part of this. And again, that's all because of you guys. So I'm going to stop there before the bell rings. Thank you.